Hi everyone, Ryan here again. In this next tutorial, we're going to focus on how to operate Dana, specifically how to use the batch processing functions, the single image functions, and we're also going to look at the kind of output that Dana gives you. Okay, so in the last tutorial, we went through and we installed all of the first part of Dana into Fiji. We have Dana 1 and Dana 2 batch here. And we also were able to explore the Dana folder a little bit and launch the dana2.exe file. Okay, so if you don't have both of these, then you're going to want to go back and watch the first video about how to install Dana. Otherwise, we're going to go ahead and proceed forward. Okay, so the first thing we're going to do now is we're going to look in our Dana folder, and we see that there is a folder titled Template Images. If you open that up, these images are all taken of human neutrophils stained with Cytox at 200x magnification. So I'm going to go ahead and select all of these images and open them up in Fiji. Okay, so we have our images here. And what I'm going to go through now and walk you through is the basic part of Dana, how to use Dana Part 1 and how to feed that output into Dana Part 2 and ultimately look at your percent NETosis, your DNA area, all the stuff that you're interested in. After we're done with that, we're going to go through and review batch processing and how that's carried out. One thing I quickly want to highlight about these images is that they're overall pretty high quality. We can see that the stain is relatively bright. We can see that the cells are pretty adequately spaced out. There's not a whole lot of overlap. And that's going to make it really easy for Dana to pick apart one cell from another and give you high quality output. So the more spaced out the cells can be while still giving you a decent number on screen, the better your data is going to be in the end. To generate our first output, we're going to go to the Plugins menu, go down, run Dana Part 1. And if we look at the output generated by Dana Part 1, what we saw it do was effectively threshold the image and then analyze all of the particles within it. And you can toggle between these yellow outlines. The yellow outlines are called ROIs, or regions of interest. You can toggle and turn those on and off by clicking the Show All button. Okay, so now that we have these regions of interest, the first thing we're going to do is make sure that these regions of interest, or ROIs, effectively capture the data that we see on screen. And one way to make this a little bit easier, it's not as big of a problem with these images because they're bright and relatively differentiated from the background, but you can go to Image, Adjust, Brightness Contrast, and if you click Auto, it's going to make the brightness between the cells, or like the regions where we see signal, and the background a lot more distinct, so easier to pick up for your eye. The next thing I'm going to do in looking at this image is make sure it does not contain too many multiples. A multiple is simply your regions of interest, or these yellow outlines, that contains more than a single cell within it. So looking at this image, I see that number 30 down here appears to be a multiple. It's a single ROI outline, and it contains what looks to be 2 cells. And while a few of these aren't a problem, and Dana's meant to kind of eliminate them, having too many can really reduce the quality that Dana Part 2 is able, the quality of data that Dana Part 2 is able to produce later on. So looking at this image, I see 1 multiple. Number 8 is that? Number 8, I think, is a single cell. So it looks like, oh, we have another multiple up here, 3. There's clearly 2 cells, and it looks okay. As a general rule of thumb, if I find more than 7 multiples in a given image, I discard it. To discard an image, I didn't really mean that you're going to delete the image. I more so meant that you're going to delete the output produced by Dana Part 1. In the earlier video, we set up our output location to be within our Dana folder. So if I navigate back to Dana, we chose to set it as this Dana1-output. So if I open that up, I see a single file, UT-U2, and that corresponds to this image right here. So if this contained more than 7 multiples, or the regions of interest did not accurately reflect the cells that we see on screen, I would just simply delete it from my output folder. Now that we understand how to delete bad images or bad output, we're going to loop through and check out some of the other images contained within the Template Images folder. So I have the output for UT-U2 here, and I'm going to exit out of this image, and then I'm going to run Dana Part 1 on this next image, That looks good. I don't really see a whole lot of multiples. It seems fine, so I'm going to keep it. I'm going to run on this other image. Okay, that one looks good too. I can toggle on and off to kind of help me see what's what. I see one multiple here, 19, but overall it seems fine. And as you do a lot of these, you get really good at picking out images that contain more than 7 multiples. This one, so 38, that's a multiple right there. 17, that's a multiple right there. 1, that's a multiple up here. So it looks like this is also going to contain less than 7 multiples, so I'll keep it. So what I'm going to go through and do now is basically do this for all of the images, and I'm going to delete any output that contains more than 7 multiples over here. What makes that a little bit easier is if you sort by date modified. So for each image that you run, it's going to output the file right to the top. So it's really easy to delete the output that corresponds to images that have over 7 multiples or do not represent the data you see on screen. Great, so we've just run Dana Part 1 on all of our images, and you can see that it's stored in our initial output folder that we selected, Dana Part 1 output, and all of the outputs are listed here. But what I'm actually going to show you is a slightly easier way to do this. So I'm just going to go ahead and delete all of these images, or all of these output files. Another feature of Dana is you can use the batch plugin mode. So if you go towards Dana 1 batch and navigate to the folder that you stored your images in, or your images of interest, in this case we're just going to do the same template images and select, Dana is going to automatically loop through and run it on all of the images within that folder. And this has the benefit of being a lot faster, but it also doesn't provide the same way to double check your images and make sure that you have less than 7 multiples per image. So this is a lot faster in the end, but it may result in slightly lower quality data, and should really only be used when you're comfortable that your images have the cells spaced out pretty neatly. Now that we have all of our output from Dana Part 1 for a single sample, and in this case a single sample corresponds to a single human subject, all of the neutrophil images from that subject, we can proceed to Dana Part 2. To run Dana Part 2, we're going to navigate back and double-click on the Dana 2 executable file that we made sure ran yesterday. The first thing we're going to do is make sure that our input and output directory are correct. So in this case, we set up the first part of Dana to output this Dana 1 output folder, and it's right now where we have all of our output files stored. So I'm going to copy the file path from that, paste it into the input directory portion here. The next part is we're going to set the output directory where we want our final output to go. Just as like a general setup, I'm just going to choose the Dana 2 output folder available in the general Dana folder. So I'm going to copy that file path and paste it in here. The rest of these parameters deal with how Dana actually runs. So this first one is the upper elimination cutoff parameter. It's going to help Dana distinguish effectively what's a multiple from what's a net in just a regular cell. We also have the lower cutoff value. That's going to help Dana exclude fragments from regular cells. We have this option to normalize on what we want to normalize relative area to. The one I recommend is the mean area of the 5 smallest non-outlier rows. And what this basically means is that of all of the cells that are not excluded, it's going to choose the 5 smallest, calculate their area, and then compute the area of every other cell compared to those smallest 5. And it does this for an entire sample, not just a specific image. You also have the option of choosing area to normalize to, so you can select the set area which all regions of interest will be computed relative to. Other parameters in Dana Part 2 include the DNA decondensation cutoff parameters, which we'll talk about more in the optimization video, and finally the net cutoff. So what we've been using is a value of 4.70, and 4.70 effectively means that if you have a relative area, or if a region of interest has a relative area greater than 4.7 times, it's going to be labeled as a net. We also have this optional parameter here at the bottom, and what the optional parameter does is it allows us to distinguish between images within a single sample. So if we look at our output files over here, you'll notice that some of them are labeled IO and others are labeled UT. The IO effectively just means that they were treated with ionomycin, and the UT means that they were untreated. Ionomycin is a calcium ionophore, and we would expect cells treated with ionomycin to have higher rates of NETosis. So I'm actually in the Dana2 output folder right now, and I have some output left over from before, but I'm just going to go ahead and delete that right now. And then I'm going to look over here. So I have all of the settings set up that I wanted. With the human cytoxane neutrophils, we've been using an upper elimination cutoff parameter of 1.5, a lower cutoff value of 20,000. We've been computing our relative area to the mean of the 5 smallest non-outlier ROIs. And we can also look at the optional parameter being ionomycin-treated images. And we've been using a net cutoff of 4.7 times. So you have the option to save these current settings if you want. I'm not going to at this time, but I'm going to go ahead and run Dana right now. Of note that when you run Dana Part 2, it's going to include all of the .csv files that are present in your input directory. So if you have .csv files from previous samples, they're going to be included in the analysis that Dana Part 2 carries out. And this is something you don't want. So you either want to delete these .csv files after you use them, or more ideally store them in a separate location for future analysis. So now we're going to look at some of the output files provided by Dana Part 2. So if we navigate to that folder, first we're going to look at the summary.csv file. So I'm going to expand this here. The summary.csv file is going to contain information on our optional parameters. So this is just the ionomycin images, on the untreated images right here, as well as both of the images combined. This first panel is going to show us the percent NETosis. So as expected, the ionomycin has higher percent NETosis than the untreated cells. Combined, we have about 14% NETosis. It's also going to show us the average area of the cells in microns. And it's also going to show us a t-test between groups. So this is the ionomycin treated versus the untreated. Additionally, Dana Part 2 also shows us the total number of cells. The higher this number, the better. It gives us a more accurate representation of our sample. The next thing we're going to look at is a output for a single image, and I'll just pick I'll pick IOR right here. Okay. So this is the output for a single image, and I'll actually open up the image that it corresponds to alongside of it. That'll probably make things easier. Okay, template images. This is IOR. Oops. I'll open an image J. Okay. And then I will run Dana Part 1 so we can see the ROIs on top of it. All right. So looking at this, each one of these numbers right here corresponds to the ROI labeled over here. We can see that there's 34, 34. And this also provides us information on things like the area in microns, the mean brightness, the min, the max brightness, circularity, integrated density, raw integrated density, which is basically the sum of all pixels in a particular ROI, the AR, the roundness, the solidity, the relative area, which is used to compute whether something's a net or not, as well as the classification. So anything that's just a regular decondensed cell and not considered a net is labeled with this X. So the first one was labeled as a regular cell, and it looks like, yeah, that doesn't look like a net at all. If we go to, okay, the first net right here, that is in ROI 7, and it looks like there's one right after it in ROI 8. It looks to be those two right here. And I would agree that those are nets. They appear pretty decondensed, they're cloud-like, they're wispy, they have a lot of tails. So I think that Dana did a good job classifying those two as nets compared to something like this, or something like this, or something like this. So if we go down the list, we have something labeled a multiple here. That's region 25. So if we look at 25, that one's right here. Yeah, that does look to be two cells, 1, 2. What else? We have another multiple labeled down here, number 30. And yeah, that's this one right here. So I would agree with that. And it looks like Dana did a pretty decent job classifying this image overall. It's pretty hard to pick up on some of them, whether it defines, whether it is actually a net or not. It's pretty close, like this one. I think that you could argue for that being a net, but Dana provides a pretty unbiased way to determine and label whether that is a net or not. The last thing that we're going to look at for this image's output is going to be these columns over here. So I'll actually expand to full screen. And this provides information on the entire image taken together. So this is the average area. This shows you the lower cutoff that you selected, as well as the number of fragments excluded, the number of multiples excluded. It shows you the percent NETosis. For this image, it was around 10%. It also shows you the number of rows that it classified as nets, as well as the number of total cells that were not excluded for being multiples or fragments. So in summary, Dana is able to provide information on DNA area in NETosis on a per cell, per image, and per sample basis. Thanks for listening. In the next video, we're going to talk more about optimizing Dana to different stains, different samples, and different cell types. Thanks.